Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will examine insect metamorphosis and focus on the two fundamental developmental pathways that insects use to transform from egg to adult. Metamorphosis in insects describes the sequence of life stages and the morphological and physiological changes that occur between those stages. Entomologists divide metamorphosis broadly into complete metamorphosis and incomplete metamorphosis. These terms are sometimes called holometaboly and hemimetaboly. Complete metamorphosis includes 4 distinct stages. First is the egg. After hatching, the organism becomes a larva. The larva grows through a series of molts and then enters a pupal stage. Finally, the adult, or imago, emerges from the pupa. Incomplete metamorphosis also begins with the egg, but the intermediate stage is called a nymph rather than a larva. The nymph generally resembles a small adult and develops through several instars by molting until it attains full adult form. One clear difference is the presence of a pupal stage in complete metamorphosis. The pupa is absent in incomplete metamorphosis, and that distinction underlies many ecological and developmental consequences. For example, the monarch butterfly transforms from caterpillar to pupa to adult, and this is a classic example of complete metamorphosis. By contrast, a grasshopper hatches as a nymph and gradually gains wings and reproductive structures over successive molts. This is an example of incomplete metamorphosis. At the tissue level, pupation involves histolysis of many larval tissues and histogenesis of adult structures. Imaginal discs in holometabolous insects are clusters of undifferentiated cells that proliferate during pupation to form adult appendages and organs. Hormonal control orchestrates these changes. Ecdysteroids trigger molting events and promote progression to the next stage. Juvenile hormone, produced by the corpora allata, maintains larval characteristics. A decline in juvenile hormone concentration at a critical time allows the transition to pupation and then to the adult form. Researchers use model species such as Drosophila to study imaginal disc development and the genetic regulation of metamorphosis. Work in fruit flies has revealed signaling pathways and transcription factors that regulate cell proliferation and differentiation during metamorphosis. Ecologically, complete metamorphosis often separates feeding niches between larva and adult. Caterpillars are herbivores that consume leaves, while adult butterflies typically feed on nectar. This separation reduces intraspecific competition for resources. Incomplete metamorphosis tends to occur in groups whose nymphs and adults occupy similar habitats and feeding niches. Because nymphs resemble adults, they can exploit the same resources and develop gradually without a pupal transformation. There are applied implications of understanding metamorphosis. Pest management can target hormonal pathways. Juvenile hormone analogs moderate development and can prevent insects from reaching reproductive adulthood. Another practical consideration is diapause. Many insects enter a dormant state during a particular stage, often the pupa in holometabolous species, to survive adverse conditions. Diapause timing is tightly linked to metamorphic control mechanisms. Evolutionarily, the emergence of complete metamorphosis is thought to have allowed major radiations in insect diversity, because larvae and adults could specialize independently. In summary, metamorphosis in insects comes in two main forms. Complete metamorphosis includes a distinct pupal stage and dramatic tissue reorganization. Incomplete metamorphosis involves gradual changes through nymphal instars without a pupa. Understanding the developmental biology and ecological consequences of these pathways helps explain insect diversity and informs approaches to control pest species. That concludes the lecture. Next session, we will examine the molecular signals that switch juvenile hormone levels during the final larval molt.
Listen again to part of the lecture, then answer the question.
For example, the monarch butterfly transforms from caterpillar to pupa to adult, and this is a classic example of complete metamorphosis.
This concludes the listening section. For more TOEFL practice tests and study materials, visit spinta.org.